Ribosomes, which are found in both plant and animal cells, are RNA and protein complex that is found in all cells. During translation, which we'll learn about with DNA, ribosomes join amino acids together to form proteins. Finally, the last one, the vacuole. The vacuole stores water and ingested food in a fluid sac. In plant cells, the central vacuole produces turgor pressure against the cell wall for cellular support. But it is found in both, it's only found in plant cells. I know normally you would watch this video by yourself, but this time I did play it in class because you've already answered all the stuff about it. I just want to make sure that you do listen to it. There's no point answering questions. I just want to make sure that you hear it. You need to take some time and fill in the plant versus animal Venn diagram. Just put the parts where they belong, whether they belong with both or just in the plant or just in the animal. Plant cells and animal cells contain different cellular structures. For example, plant cells have chloroplast, while animal cells do not. Most plant cells possess a cell wall and chloroplast. Animal cells never contain these structures. Both plant and animal cells contain vacuoles, but those found in animal cells are much smaller. Animal cells have centrioles and lysosomes, which aren't found in plant cells. Centrioles play a major role in organizing the mitotic spindle during cell division. Lysosomes contain enzymes which digest food particles, excess or old organelles, and engulfed foreign particles such as viruses or bacteria. All cells are surrounded by some type of membrane. Plant cells have a cell membrane and a cell wall, while animal cells only have a cell membrane. It is the lack of a cell wall in animal cells that allowed animals to develop greater diversity of cell types, tissues, and organs. It is important to note that all cells do not always have all the organelles. Even within plants, many cells do not contain chloroplast. Cells located in the roots are not exposed to sunlight and do not photosynthesize. Therefore, chloroplasts are not necessary. The same is true for cells that compose animals and all other complex organisms. Cells within multicellular organisms are highly specialized for the specific function they perform. Prokaryotes versus eukaryotes. Prokaryotes do not have a true nucleus or other membrane-bound organelles. Prokaryotic organisms differ from eukaryotic organisms in complexity and structure. Most prokaryotic organisms are unicellular or single-celled. All prokaryotes lack a well-defined nucleus and are much smaller and simpler than eukaryotic organisms. Although prokaryotes do not have genetic material, DNA, their DNA is not separated from the rest of the cell by a nuclear membrane. Also, prokaryotic DNA is not arranged in chromosomes, and it tends to be circular rather than linear. Prokaryotic cells do not contain mitochondria, but they get their energy from sunlight or chemicals in the environment. They have simpler stages of growth and development. They are simple organisms such as bacteria, blue-green algae, and archaea are examples of prokaryotes. There are many differences between eukaryotes and prokaryotes. Prokaryotes are far simpler and smaller than eukaryotic cells. Fill in the chart below comparing the two using we have learned so far. You need to read the article or the 2 paragraphs in your note packet and answer the questions as to which ones relate to what. I'm not going to give you the answers on the video, but there's a good chance that we will go over those in class, and possibly I'll give the answers then. These are the answers to the second part of the school where it starts with the security system, the principal, and what all's going on in that activity. The cell membrane. The main purpose of the cell membrane is to control the movement of substances in and out of the cell. Another name for the cell membrane is the plasma membrane. Both animal and plant cells have cell membranes. In animal cells, the cell membrane separates the cell's internal environment from its external environment. In plant cells, the cell membrane is surrounded by a cell wall. The lipid bilayer. This should be a review because we basically covered this in the second unit. The proteins and lipids in the cell membrane form a lipid bilayer that includes phospholipids. In the lipid bilayer, we have the hydrophilic, which is the water-loving heads, and of the phospholipids face outward, and the hydrophobic water-fearing tails face inward. This arrangement allows the cell membrane to control the movement of substances in and out of the cell. In fact, due to this arrangement, polar molecules are unable to go across the cell membrane unless they pass through channels, or their diffusion must be facilitated by carrier proteins. Cell transport. In order for cells to function properly, they must be able to transport materials such as water and nutrients in through their cell membranes, and they must be able to transport materials such as waste and excess substances out through their cell membranes. Materials can enter and exit through the cell membrane by passive transport or active transport. Passive transport is a type of cell transport that does not require the cell to expend energy. Diffusion, facilitated diffusion, and osmosis are types of passive transport. Diffusion, or simple diffusion, involves the movement of material along a concentration gradient. That is, materials move across the cell membrane from an area of higher concentration to an area of lower concentration. Diffusion will continue across a semi-permeable membrane until particles are equally concentrated on both sides of the membrane. Small molecules such as oxygen, ethanol, and carbon dioxide easily diffuse across the membrane. Some molecules cannot simply diffuse across the cell membrane because they are too large or because they have a chemical structure that prevents them from passing directly through the cell membrane. Facilitated diffusion is a type of diffusion that uses special transport proteins to transfer molecules across the cell membranes. Like diffusion, facilitated diffusion involves the movement of material along a concentrated gradient, so no energy is required. Glucose is an example of a molecule that is transported by facilitated diffusion. Osmosis is the type of diffusion that specifically involves the movement of water across a semi-permeable membrane. During osmosis, water moves from areas of high concentration to areas of low concentration, so energy is not expended. Cells react differently when placed in solutions with different concentrations. The effects of osmosis on cells. When you have an isotonic cell, the concentration of solutes is the same inside and outside the cell. Water molecules move equally in both directions. Hypertonic, the solution has a higher solute concentration than the cell. A net movement of water molecules out of the cell causes it to shrink or shrivel up. Hypotonic, the solution has a lower solute concentration than the cell. A net movement of water molecules into the cell causes it to swell like a balloon. Below are animal cells placed in beakers of various concentrations. Draw an arrow to show which way the water would move by osmosis. Fill in any missing percentages for the water or solute, and identify the type of solution, so whether if it's isotonic, hypertonic, or hypotonic. Now you need to answer the questions on passive transport. I'm not going over these on the video. I will go over them in class. Active transport is a type of cell transport that requires the cell to expend energy. Active transport typically occurs when substances try to move against a concentration gradient. For example, they form an area of low concentration to an area of high concentration. or when molecules are too large to undergo facilitated diffusion. Cell membrane pumps, endocytosis, and exocytosis are types of active transport. Cell membrane pumps are protein molecules that are embedded in the cell membrane. These molecules use energy to pump substances across the cell membrane against the concentration gradient. The sodium-potassium pump is an example of a cell membrane pump. The sodium-potassium pump is found in almost all animal cells, and it plays a vital role in transporting nerve impulses. Endocytosis is a process through which large molecules are transported into a cell. Into. During endocytosis, the cell membrane surrounds a molecule and forms a vesicle. The vesicle then pinches off of the cell membrane, so its contents can be released inside of the cell. Exocytosis is a process through which large molecules are transported out of the cell. They're exiting out. During exocytosis, a molecule is packaged into a vesicle. Then the vesicle fuses with the cell membrane and expels its contents outside of the cell. Using the pictures I have provided for you, I want you to cut them out and make your own note cards. This will be an easy study guide for you to study for your test. This finishes up the unit on cell structure and function and the cell theory. Next in your packet is going to be some questions like you always have. And then there are questions that go along with pictures to color. So as you answer the question, you're supposed to color that part of the cell that color. There's quite a few of those, and then the pictures are behind them. We'll probably do these in class and I'll go over them. Then there is a study guide behind that, having you identify the parts of the cell. I think there are 50 questions. And then you have, of course, your edible cell project. This unit went a lot faster than I thought it was going to go, but that's okay. We'll be good on time. Thanks.